welcome back. My name is Dr. Scully. I'm going to be giving you the third lecture in our series of human kidney physiology. We started off thinking about body fluid compartments and the composition of those compartments, and then we moved on to the very first function of the kidney, which is glomerular filtration rate, recognizing that now the filters in our kidneys present the kidney tubule with a large volume of water and a large number of solutes. So now we're going to talk about how we can begin to link tubular function to the glomerular filtration rate. So let's start off with a case of the day, which is called an unexpected finding, and we'll think about this in relationship to our previous patient in lecture 2. This is a 15-year-old who goes to have a routine physical examination by his family doctor that includes a urinalysis. Now, urinalysis is performed with what's called a urinary dipstick, and that means that you take a urine sample, you dip the stick in, and it has actually a series of measures that it can make, and one of those is glucose. So a urine dipstick test, the urinalysis, showed that blood and protein were absent in the kidney urine sample, but the sugar or glucose was detected. So the family doctor appropriately did an urgent blood glucose lab measurement and found that it was 4.5 millimoles per liter. And that's normal level. So typically we would expect to see between 4 and 10 millimoles per liter depending on recent meals and activity. So repeated urinalyses showed that glucose was present when there was a normal blood sugar level. And the question the family doctor has for us, based on our understanding of renal physiology, is, does this patient have diabetes? So let's see if we can review some aspects of kidney tubule function and begin to address kidney handling of a small solute like glucose and how we can use that knowledge to interpret these laboratory findings and determine whether indeed the patient is diabetic. So, tubular function is all about transport of water and transport of solutes. So just a few general comments, then we'll sort of focus on tubular physiology and water very briefly, and then tubular physiology and sodium using the extracellular fluid, most common solute, sodium. And then we'll take a detailed dive into each of the tubular segments, because he's going to see that the nephron, the next stage the ultrafiltrate encounters, has a very complicated anatomy. And then we're going to focus on glucose to close and see if we can understand glucose and how the kidney handles glucose. Now, one of the important things to remember here is that the anatomy of the kidney is very difficult to separate from the physiology, and we'll try to emphasize that principle as we move forward. So here we are back to this kind of organizing notion. Again, body fluid compartments flipped on end so that we have a plasma volume. The plasma volume is delivered to the kidneys, into the kidney, and we create an ultrafiltrate. So remember that the kidney glomerulus, that is the 1 to 2 million, filter approximately 150 to 170 liters a day. It's kind of interesting to think that in a 1-hour lecture, which is of course 1/24th of a day, probably close to 6 or 7 liters is filtered by your glomeruli, and that volume is presented to what we'll call the tubular compartment within the kidney. Now, when we first focused last time, we talked about the arterioles coming up, the arcuate arteries, the intralobar arteries, the afferent arteria, and all these filters. And here's the orientation of the nephron. Now you can see that it looks very much in this picture like spaghetti. And sometimes that's the way I think about it. It's a tubule that has an orientation. At the top, the glomeruli, and then a vertical orientation. I often imagine that the complex orientation of the nephron is actually like pins in a pin cushion. They all protrude out, and at the surface of the top with the glomeruli, and then the nephron functions start at the surface of the kidney, then work their way down, and eventually lead to the formation of urine. Well, let's take a look at that anatomy a little bit. So here's our glomerulus, and here's our afferent arteriole, here's our efferent arteriole, and within this capillary loop is a ultrafiltration will occur on the basis of Starling forces, and indeed it's going to put some 150 liters into the lumen of the glomerulus, and this is an example of a single nephron. So each glomerulus that we have, the 1 to 2 million, is attached directly to a nephron. And we taught these are the nephron segments. So the very first part is called the proximal tubule. And then there's a loop of Henle. Again, thinking of these arranged vertically from the surface of the kidney like a pin cushion, in which at the top of the glomeruli and proximal tubules, and then the loop of Henle does a deep dive towards the interior of the kidney, and then it ascends to form the distal tubule, and then finally the collecting duct. And then this is where the final urine flow proceeds on to the bladder and for excretion. So the nephron has several important segments: the proximal tubule, the loop of Henle, the distal tubule, and the collecting duct. And each of these segments of the kidney nephron is presented with a large volume of water and a large number of solutes, and it is the processing, if you like, of that ultrafiltrate volume and solute composition that allows the kidney to regulate the volumes of our our body fluid compartments and the composition of those compartments. Now, if I cut through the cortex of a kidney, you can see these are the filters, these small microscopic structures. Those are the glomeruli, and you can see a lot of these open spaces, and those open spaces represent the lumen of the kidney tubule. So here's a kidney tubule here, and a light micrograph showing, and here's the lumen of that kidney, and this is it. These are representing mainly proximal tubules up here, and this is a cross-section of a proximal tubule. And if we take a colorized electron micrograph in the third image here, we begin to better understand the structure of the tubule. So we know this represents the basement membrane, this represents the luminal membrane, and this represents up here the interstitial compartment. So what we have is a epithelial cell that is actually polarized. That is, it has different structural orientation. It has a brush border, particularly in the proximal tubule, the cell body, and then it has a basement membrane, and opposite the basement membrane, a tubule-interstitial compartment. And you can imagine that what this tubule is going to be doing is reabsorbing and indeed sometimes secreting constituents into the lumen as it processes that ultrafiltrate and regulates the passage of solutes so that it maintains body homeostasis. So if we think about it in sort of simple terms, what does the kidney do? Well, it filters a large volume of plasma, water, and solutes. That's the glomerular ultrafiltrate, the GFR. And the filtered fluid then enters the kidney tubule lumen. And the tubules reabsorb solute and water and or secrete solutes so that despite what we eat and drink, body composition and body fluid volumes remain relatively normal. We're going to talk in general terms about the functional elements within each of the nephron segments today, and then in subsequent lectures we're going to talk about how those segmental functions are regulated to do, in fact, the third bullet here, which is to maintain composition and volume. So, tubular physiology, water. So, the first thing again is that normal, it's just some quantitative numbers. The normal glomerular filtration rate is equivalent to 150 liters a day. So it's actually, this should say greater than 100 mils per minute, or 150 liters a day. And your normal urine volume varies depending upon what you drink, but varies from about half a liter to about 2 liters a day on average. And that depends again on how much water you drink. So one of the remarkable things to realize is that 99% or more of the filtered water that enters Bowman space and then enters the tubule lumen is reabsorbed by the time that luminal flow gets to the end of the collecting duct. So one of the remarkable things is the kidney goes through a lot of work to collate a large volume of ultrafiltrate, and then it reabsorbs almost all of it. Teleologically, I've often wondered, why would it do that? Because it's an energy-requiring function to reabsorb water, as we'll see, but it's probably related to the fact that it allows the kidney to secrete the metabolic waste and at the same time preserve water, because you and I evolved in an environment where there wasn't a lot of environmental water, and so we don't want to lose water. And this also explains why, if you filter 150 liters a day, remember that's 5 times your total body water, the reabsorption of that water means that normal kidney function, while filtering large volumes, retains those volumes and limits changes in your total body water, and indeed regulates those changes. So let's look at this in a sort of quantitative way and think about how this might look in terms of the different numbers we have to consider. So just going to start, here's our glomerulus. Now, we start off with the heart and the cardiac output, the renal blood flow and the renal plasma flow. And the renal plasma flow comes into here, and about 20% of this renal plasma flow every minute, about 1/5, is filtered into the kidney tubule, and this represents the kidney tubule, not divided into the structural elements that I showed you earlier, but just that sort of generic, as if we took that complicated anatomical organization of the nephron and kind of stretched it out, sort of like we had a bundle of a hose in your backyard, and then you pull it out and stretch it out into a linear structure. Nephron is not linear, but indeed for this representation. And then as that ultrafiltrate moves, 20% of the volume is filtered, but 19% of that 20% is reabsorbed, and less than 1% is excreted at the end of the day. So these are remarkable numbers, so that you and I present less than 1% of the filtered load to our bladders that we then excrete. So these are important representation, again, of how water moves. And we're going to talk not so much about water today, but how water is handled in the different segments, and critically in a couple of lectures, how that water handling is regulated. So what about the sodium? So the normal GFR, and again I have the same mistake, is 100 mils per minute or 150 liters a day. That contains 22,500 millimoles of sodium. And yet, you and I, on a normal North American diet, probably take in about 150 to 200 millimoles of sodium. And so, again, 99% of the filtered sodium is reabsorbed by the kidney each day. We would be in extraordinarily tough times if the kidney didn't reabsorb that sodium, because sodium is not common in the environment in which we evolved. Once we, from an evolutionary point of view, once we crawled out of the primordial sea, there was not a lot of salt around, and so the kidney has that ability to filter large volumes, process the urine to excrete metabolic waste, but at the same time conserve water and conserve sodium. Now, how do the kidney epithelial cells reabsorb sodium? The first thing to realize, and we pointed it out a little bit on that colorized electron micrograph, is that the kidney epithelial cells are polarized. That is, they have a structure, a lumen, and a basolateral membrane. And that structure, that polarized nature, allows for directional transport of solutes and water. And different transport proteins are present in the luminal membrane and the basolateral membrane. So if we take that kidney tubule cell, here's the brush border, here's the nucleus, here's the basement membrane. This represents that polarized area in which there are certain transporters here and certain transporters there. What's critical about the polarized epithelial cell in the kidney is that sodium-potassium ATPase, which we talked about in lecture 1, which was related to the ability of you and I to maintain high intracellular potassium, low intracellular sodium, and the opposite in the extracellular space, presents in the basolateral membrane. So sodium-potassium ATPase in the basolateral membrane and different transporters in the proximal tubule allow for the so-called vectoral transport of salt and water. So here's a cartoon. So here's our colorized electron micrograph. Here's the basolateral membrane, and here's the luminal membrane. Here's showing a brush border. So again, here's the interstitial space, and this is the tubular lumen. So it's kind of like we've taken a picture here. This represents our epithelium, and there are junctions and proteins that maintain adherence junctions between these cells. And here is our membrane, and here's where sodium is actually filtered in high quantities, 22,500 millimoles a day. So how does the kidney reclaim sodium from the tubular lumen to the interstitial space? It does so with a family of luminal sodium transporters. And all of these luminal sodium transporters are critically dependent on the basolateral sodium-potassium ATPase that critically maintains a low intracellular sodium concentration. So the key ability of the polarized epithelial cell to absorb sodium from the lumen and transport it to the interstitium is critically dependent on sodium-potassium ATPase, and therefore critically dependent on the generation of ATP in proximal tubule cells, for example. So none of you would be surprised if I showed you an even higher powered electron micrograph that showed that each of those proximal tubule cells has a tremendous number of mitochondria in it, because the generation of ATP to maintain that low intracellular concentration is critical. So the high filtered load of sodium comes into contact with the luminal membrane of the proximal tubule cell. And this shows one type of transporter, and this is an anti-transporter, and this is called a sodium-hydrogen exchange. There are a family of these proteins, and this is represented as a luminal transport protein, this red circle. And it has a function. It moves sodium into the cell, down a concentration gradient from the luminal compartment into the cell, and it exchanges it for hydrogen ion. So one class of transporters are anti-transporters, in which the gradient between sodium here and here drives the reabsorption of sodium in partnership with or exchange with the movement of hydrogen into the lumen. We're going to come back to this because the kidney uses this to maintain acid-base balance. So antiporters that are retained within the luminal membrane use the low sodium concentration to drive sodium reabsorption and drive an exchange, in this particular example, with hydrogen ion. If we were to ablate sodium-potassium ATPase activity, then the sodium concentration couldn't be maintained low, and that would impair the kidney's ability to reabsorb sodium using this type of transporter. So remember, the sodium-hydrogen exchanger plays a key role in sodium reclamation. The sodium enters the cell and then is promptly pumped out by sodium-potassium ATPase. So here's another kind of transporter. Again, the key thing to remember is that ATP-driven sodium-potassium exchange in the basolateral membrane maintains a low sodium concentration, providing the concentration gradient for sodium reabsorption, which is high in the luminal compartment. And that sodium moves down a concentration gradient, and in this particular case, there's a co-transporter. So it's the gradient of sodium that drives the movement of sodium from the lumen into the cell. And if you like, glucose or phosphate, hop onto that transporter and utilize the gradient in sodium to enter the cell. So unlike the antiporters, which exchanged, in that particular case, positively charged solutes, in this particular case, the sodium positively charged is associated with the transport of another small solute, glucose, phosphate, amino acids. There's a whole family of transporters in the luminal membrane to reclaim constituents of the ultrafiltrate that the kidney doesn't want to excrete. And that includes, in this particular example, and this SGLTs stand for sodium glucose transporters. So this could be a sodium glucose transporter in which the gradient of sodium drives the reabsorption of glucose. Alternatively, sodium phosphate transporters in which the gradient of sodium drives the reabsorption of sodium and phosphate, and then subsequent entry into the interstitial space. So antiporter protein, transport proteins that reside within the luminal membrane, and co-transport proteins that reside within the luminal membrane all drive sodium reabsorption with other solutes involved, and the low sodium is always critically dependent on the basolateral sodium-potassium ATPase, and therefore cell function, of course, is critically dependent upon the generation of ATP. Now, another kind of protein can exist within the luminal membrane, and this is the one we'll talk a lot about when we talk about the regulation of sodium reabsorption and with the regulation of potassium excretion. And this is an epithelial sodium channel, and I've represented it. It has multiple components. I've represented it as two round circles in the luminal membrane. This is a channel that allows sodium to move across the luminal membrane down a concentration gradient. Epithelial sodium channel, ENaC for short, is a famous transport protein that plays a key role in potassium secretion and sodium reabsorption. Another type of luminal membrane transporter that shares the same features as the cotransporters and the antiporters. They're all dependent on a low intracellular sodium concentration, which is dependent on a functioning sodium-potassium ATPase that is restricted to the basolateral membrane adjacent to the interstitium. So again, sodium in the lumen in high concentrations moves down a concentration gradient and then is summarily pumped into the interstitium. Actually, this process, as you take a solute from the lumen and move it into the interstitium, you affect the osmolality. And then that affects the reabsorption of water. So water moves through that cell through aquaporin channels, and it also moves, depending upon permeability, through paracellular. So by transporting a solute, like sodium, from the lumen into the interstitium, from the lumen to the cell to the interstitium, you create osmolar gradients, and water follows the osmolar gradient, so that you reabsorb both salt and water in parallel in all of those segments, in the proximal tubule particularly. A little bit different in this cell, which we'll come back to. So as a model of tubular reabsorption secretion, what do we want to think about? Well, critically, the low intracellular sodium concentration is driven by sodium-potassium ATPase. That's a ubiquitous protein that maintains a low intracellular sodium concentration, a high intracellular potassium concentration in all the cells of our body, but in polarized epithelium that sit on a basement membrane, and which there's restriction of those transporters to either a basolateral or luminal membrane, it allows transport in a vectoral or directional relationship. So luminal sodium enters the cell down a large electrochemical gradient. We'll come back to the cell interior negative, but for now, we're really thinking about the chemical or concentration gradient. And then the sodium that enters the cell exits the cell at the basolateral membrane. So polarization, specific localization of transporters leads to the directional transport of sodium. This is a dominant process in the proximal tubule, and it leads to the reclamation of sodium, and water follows the sodium. So reabsorption of secretion of other solutes, importantly, is linked to sodium. It takes advantage of the fact that sodium-potassium ATPase renders the interior of the cell, renders the sodium concentration in the interior of the cell low enough to drive a concentration gradient. And that energy derived from that concentration gradient is what the transporters use to reclaim things like glucose, amino acids, and phosphate. So let's take a detailed look at the tubular segments. We recognize that, of course, these segments will have different kidney functions, and although I've stretched them out to be linear when we talked about water, we recognize that different segments have different luminal transporters and different water permeabilities and different functions. And so let's walk through that. So how, we might ask ourselves, how do nephron segments differ? So again, here's our nephron. First function, glomerulus. We create an ultrafiltrate that courses through that nephron. First seeing the proximal tubule, then the loop of Henle, then the distal tubule, and then the collecting duct. And in a sequential manner leads to sodium reabsorption and water reabsorption together with other constituent solutes. Specific luminal transport proteins differ in these segments, and we're going to go over that in a minute. The leakiness to solutes and to water differs. So the ability, for example, of potassium to move across a membrane, a segment of the nephron, the ability of water to move across differs. The tight junctions between cells confer those differences in solute and water permeability in part. And then the presence of aquaporin channels within the luminal and basolateral membranes also facilitate, or not, the movement of water across those segments. And we're going to talk particularly in the collecting duct about how the regulation of water channels influences cell permeability to water. And then there's going to be a host of hormones that influence the transport characteristics of these membrane segments, including solutes and water. And those include aldosterone, vasopressin, and angiotensin II. And we're going to return to each of these when we talk about the regulation of sodium and water reabsorption. But these are the ways in which a nephron segment can differ. Transport proteins, leakiness, tight junction biology, aquaporin channels, location and function, and then the presence of hormones that respond, the presence of hormone receptors that respond to those ligands and influence the biology. Remembering, of course, aldosterone is a steroid hormone, and has intracellular receptors, and vasopressin and angiotensin II are small peptides that respond to G protein-coupled receptors that are differentially expressed in segments of the nephron. So let's look at the proximal tubule. Proximal tubule is the workhorse of the tubular segments in terms of the bulk of water and sodium reabsorption. Sodium, chloride, water, potassium, and bicarbonate are all reabsorbed in the proximal tubule. Indeed, we'll find out that about 2/3 of all the water reabsorption and sodium reabsorption occurs in the proximal tubule. The most important luminal transport protein that influences sodium reclamation from the lumen and influences secondarily water movement from the lumen through and between the cells into the interstitium is actually the sodium hydrogen exchanger. And it's this time I've labeled it NEH3 for sodium hydrogen exchanger 3 because they're a family of these proteins. Critically sitting in the luminal membrane and critically taking advantage of the low intracellular sodium concentration so that sodium moves down the concentration gradient, water follows. Important for bicarbonate reabsorption. We're going to spend a good chunk of the lecture down the way talking about how important that is. But critically, it's the site of glucose, phosphate, and amino acid transporters that are only found in the proximal tubule. That allows the kidney to reclaim those solutes from the ultrafiltrate and to play some role in regulating their excretion, but particularly things like glucose and amino acids, so proteins and carbohydrates, are ones that the kidney wants to preserve and doesn't want to excrete. We don't want to excrete a carbohydrate because we have to replace that from the environment. And we don't want to excrete amino acids because we have to replace that from the environment. So the kidney plays key roles in making sure that the urine doesn't waste these metabolites. And then very leaky. That's the critical, it's that leakiness of the proximal tubule. That means as sodium is reabsorbed down concentration gradients and then pumped by sodium potassium ATPase into the interstitium, the changes in osmolality across those membranes from the luminal membrane and the basolateral membrane allow water to move readily along that osmolar gradient, so that as sodium is reabsorbed, water is reabsorbed. And actually, if you and I were sitting in a very proximal tubule and the very distal proximal tubule, we'd find that the osmolality was the same. And that's because sodium and water are moved in tandem and don't influence the osmolality within the lumen. That's not the case later on in the nephron. Thick ascending loop of Henle. Well, here, if we said the proximal tubule was where the bulk were some 60 to 65%, here about 20 to 30% of the filtered sodium is reabsorbed. There's differential water permeabilities, and we'll come to that when we talk about water regulation. What's the key protein for water sodium reabsorption in the thick ascending limb? It's a luminal transport protein. It's a co-transporter. But instead of just being sodium plus one other thing like phosphate or glucose, it's a sodium potassium 2-chloride transport protein. That means all four of those solutes have to dock on that protein before they're translocated from the lumen into the cellular compartment. Sodium potassium 2-chloride, so NKCC2. This is an important protein to remember because we use it a lot in clinical medicine. Maybe I shouldn't say we use it, we abuse it by using a drug called furosemide, which displaces one of the chlorides in the transporter and limits the ability of that transporter to move the solutes from the lumen into the cell, and therefore inhibits its activity. A consequence then you might predict of giving someone furosemide, which is a diuretic as listed here, is to cause more salt and water to remain within the lumen of the nephron. The thick ascending limb, the ascending limb is very impermeable to water. The descending limb is very permeable to water. The descending limb doesn't transport much sodium. The ascending limb transports sodium. So this interesting architecture of this loop, which is contiguous, is associated with different water permeabilities. As you and I are riding from a leaky epithelium in the proximal tubule, we enter the descending limb where there's not this transporters taking sodium out of the lumen anymore, but rather what we're seeing is water permeability. It's leaky. And then we turn the corner here at the loop of Henle and go in the ascending limb, and all of a sudden everything changes. The water permeability goes down, so water can't move out. But sodium transporters appear again and move sodium out of the lumen in the thick ascending limb. Fluid ascending, leaving the thick ascending limb is always hypotonic. So here we said the osmolality was normal, or 280, here 280. Here it can be 70 to 100, because actually leaky tubule epithelium, reabsorption of salt and water in tandem. Here, the sodium reabsorption occurs, but the collecting duct, or the ascending limb of the thick loop of Henle, not permeable to water. This function of the loop of Henle, both its anatomy and the differential sodium and water transport and permeability characteristics, lead to a concentration in the medulla that we'll talk about in detail, because it enables you and I to concentrate our urine. And we'll see how this anatomy, coupled with this very particular physiology and transport proteins and aquaporins, enables the kidney to create an interstitial milieu that allows you and I to concentrate our urine if required. The distal convoluted tubule. So we've raced through the proximal tubule, we've come to the loop of Henle, we've gone up the thick ascending limb, and now we're sitting in the distal tubule. 2/3 of the water another third of the, well, let's say 60%, another 30%. Now we get down here, the distal tubule, there's not, almost all of the sodium and water has been reabsorbed, but the distal tubule is starting to fine tune the constituents, the solutes and water that are in the lumen of the nephron. It reabsorbs about 10% of the filtered load of sodium and water. And here, the dominant sodium transporter is different. Proximal tubule, it was sodium hydrogen exchanger 3. Thick ascending limb, sodium potassium 2 chloride cotransporter. The distal tubule, it's a sodium misspelled chloride cotransporter, and it moves sodium and chloride into the cell. Thiazides are a very common drugs that are used to treat hypertension, and thiazide diuretics influence the sodium chloride cotransporter in the thick ascending limb. So furosemide causes sodium loss in the nephron by inhibiting transport here, and thiazide diuretics cause sodium loss in the nephron by inhibiting sodium chloride cotransport activity within the distal convoluted tubule. This segment of the nephron is important in reducing and maintaining the concentration of sodium within the lumen, so that by the time we get to the collecting duct, we can finalize the concentration depending on the physiologic stimuli that the kidney is observing. Now, the collecting duct, we're going to spend a lot of time kind of wandering about the collecting duct because it plays a key role in so many aspects of renal physiology. Now, only 1 to 3% of the filtered sodium load is reabsorbed in the collecting duct. So it's really doing that final fine-tuning. Bulk here, here, to a lesser extent here, and a lesser extent here. The sodium transport protein is different. It's the famous ENaC that we talked about. So again, proximal tubule, sodium and hydrogen exchanger 3. Thick ascending limb of the loop of Henle, sodium potassium 2 chloride cotransporter. Distal tubule, sodium chloride cotransporter. Collecting duct, the epithelial sodium channel. What's key here is that they're all aldosterone receptors, so the hormone receptors, and that these hormones, the aldosterone, the mineralocorticoid, have a key impact on the sodium potassium ATPase channels and can regulate sodium reabsorption. They also have receptors for vasopressin and therefore can influence water permeability. This segment of the nephron is not permeable to water unless vasopressin is present. It also plays a critical role in potassium secretion. So if you and I were here in the proximal tubule and then here at the end of the proximal tubule, we'd find potassium. And then as we went around here and then traveled up through the thick ascending limb, the potassium would virtually disappear. There'd be very little potassium here because sodium potassium 2 chloride pulls it out. And then as we round the bend here and come into the collecting duct is responsible for potassium secretion passively through luminal potassium channels, and we'll talk about that. So it has a low capacity for transport, but a very important ability to create gradients that serve to influence either sodium reabsorption, potassium secretion, and water reabsorption. So let's finish up based on those general comments by talking about a small solute glucose so we can come back to our clinical case. So let's take a careful look at glucose reabsorption by the kidney. Now remember, from a teleology perspective, it makes a lot of sense that the kidney would want to reclaim glucose that was filtered by the glomerulus. Glucose is small solute. It will enter Bowman's capsule without any restriction. If the kidney wasn't set up to handle reabsorption of glucose, then you would spill that carbohydrate in your urine and you'd have to find it in the environment. So every calorie you lost in your urine, you'd have to look around for a calorie in your environment. So it makes sense from an evolutionary efficiency perspective to reabsorb the glucose that filters into Bowman's capsule in normal physiology. Now, here on the y-axis is the filtration rate of glucose. And here on the x-axis is the plasma glucose concentration. Now, in this particular case, this is in American units taken from your textbook. This is milligrams per deciliter. Think this is equivalent to about 5 to 6 this is in Canada we would say millimoles per liter, and 100 is about 5 to 6 millimoles of glucose. 300 is 18, 500 is about 30. So as the plasma glucose rises, the filtration of glucose will rise in parallel. Why? Because if you and I are sitting in the lumen of a glomerular capillary, the small solute glucose concentration rises. As ultrafiltration occurs, the glucose will be pulled into Bowman's space. And so you and I will be sitting in the capillary and we'll say, gee, the concentration of glucose in the plasma is up, and then we'd be sitting in Bowman's space and we'd say, oh yeah, the concentration of glucose is up. Why? Because as water is entrained, moves on the basis of Starling forces into Bowman's space, it pulls these small solutes in an equal concentration to what the concentration was within the capillary lumen. So as the concentration rises, the filtered load of glucose will rise in parallel. Now, what about reabsorption? Now, reabsorption is dependent upon luminal membrane proteins that co-transport sodium and glucose. And therefore, the reabsorption of glucose in the proximal tubule is dependent upon that low intracellular sodium concentration, which of course is dependent upon the basolateral membrane sodium-potassium ATPase. So now let's look at the reabsorption of glucose, not the filtered load, the reabsorption. And then let's look at the plasma glucose on the x-axis. So as the plasma glucose rises, the reabsorption rate rises, so that the reabsorption is in parallel. So that means that as glucose rises in this particular example, say from 5 or 6 to 18 millimoles per liter, the kidney proximal tubule cell is able to reabsorb that glucose until you reach a maximum, a transport maximum termed Tm. And then after that, the reabsorption of sodium doesn't keep pace. So that you reach a maximum ability to reabsorb. You're really a physical limitation based on the based on the tubular flow rates and the sodium co-transporters, sodium glucose co-transporters that are able to reclaim that sodium. So we get a reabsorption that rises in parallel with the filtered load and then reaches a maximum and remains constant. This is termed the renal threshold of about this is maybe a little bit high, but about 18 millimoles per liter. So this looks not at the filtered load, not at the reabsorption, but rather at the excretion, the loss of glucose into the urine at the end of the day. And as the blood glucose level rises, there's no excretion of sodium until you reach about 300 milligrams per deciliter in this example. And that's because the kidney in this range is able to reabsorb sodium and glucose, or at least able to reabsorb glucose and co-transport with the sodium. But after this threshold is reached in which the transport maximum is exceeded, there's a progressive increase in glucose excretion. So in someone whose blood glucose levels rise, the kidney is able to compensate by reabsorbing that glucose load until it reaches a point of maximal transport capacity. And at that point, glucose begins to spill into the urine. So this puts all three together. Here's the filtration, reabsorption, and excretion illustrated on the y-axis with the plasma glucose on the x-axis. The red dots represent the increasing filtered load as plasma glucose goes up. The green line represents the increased transport into the peritubular capillaries in parallel until the transportation is maximized. And then once the transport maximizes, if the glucose concentration continues to rise, there'll be a parallel increase in glucose excretion in the urine. Now, how does that happen? Well, it turns out sodium glucose cotransporters are amongst the most studied transport proteins, and indeed one of the most exciting medicines in clinical use today is a group of drugs that inhibit this transporter. Well, that's a story for another day. So, critical, these sodium glucose cotransporters, they save you and I from entering an environment in which we'd have to find an extra 600 calories a day. The urine is normally glucose free. Why? Because the normal glucose concentration is down here, and that allows the kidney to reabsorb all the filtered load of glucose. There are two transporters, two sodium glucose transporters. One is more proximal to the other, and it's a low affinity, high capacity. It's kind of like a mop, but it has a tremendous capacity to reabsorb glucose, but a low affinity means that glucose can still escape. So if you imagine as you're moving down the lumen of the proximal tubule, you have these mops that mop up most of the glucose, and after you exit to the more distal aspects of the proximal tubule, have these high affinity transporters, lower capacity. They don't have to have a high capacity, but they have to have a high affinity to mop up the last of that glucose within the lumen of the kidney tubule. So let's return to the case. 15-year-old had a routine exam. Blood and protein on a urinalysis were negative, so that didn't indicate that there was any kidney problem, but glucose was positive. So the first thing the physician appropriately asked was, well, what's the blood glucose level? Because if there's glucose in the urine, that's not normal. And I expect the blood glucose level to at least exceed 15 to 18 millimoles, 10 to 15 millimoles per liter in order for glucose to appear. So the blood glucose was normal, which is a surprise. So repeated urinalyses show glucose with a normal blood sugar. So how do we reconcile that observation? First, we know that the patient doesn't have diabetes because the blood glucose levels are normal. And we anticipate that the kidney ought to be able to reabsorb all of the blood glucose. But how come it appears in the urine? Well, in this particular case, so the patient's blood glucose is normal, he should be she should be reabsorbing all of that glucose. We're not sitting out here in that area. So why isn't that occur? Well, this young man has a what's termed renal glycosuria. He was subsequently found to have an inherited mutation in the sodium glucose cotransporter 2 gene. And that led to a loss of function of the SGLT2 protein. Now, what that means is that he didn't have the ability to capture glucose in the lumen of the nephron in partnership with sodium. So although the sodium gradient for reabsorption of sodium remained, there was no sodium glucose 2 transporter. As a consequence, he spilled glucose into the urine. So no diabetes, but a genetic defect in sodium glucose cotransport. And actually, I was mentioning inhibitors of these drugs are now being found to have a number of actually beneficial effects, although they interfere with normal renal physiology. So, summary. Reabsorption and secretion of most solutes by tubules is linked to sodium-potassium ATPase, that integral membrane protein that we talked about in Lecture 1. Sodium crosses the luminal membrane down an electrochemical gradient. That gradient created by sodium-potassium ATPase provides the energy for the reclamation of other small solutes from the lumen of in the proximal tubule particularly, but also for the reabsorption of sodium in other segments. Luminal sodium transport can occur with cotransporters, antiporters, and with sodium channels. Bulk reabsorption of sodium, water, chloride, and bicarbonate is dominated by the proximal tubule and the thick ascending limb of the loop of Henle. Regulation of the final concentrations of sodium, water, potassium is actually a function of the collecting duct, and we'll learn a lot more about that. The proximal tubule, leaky, allows lots of water reabsorption to occur as sodium is reabsorbed. The collecting duct, impermeant to water unless it sees unless in the presence of vasopressin. And organic solute transporters, which play a role in the reclamation of amino acids and other organic metabolites, are all present in the proximal tubule. We haven't talked too much about that. We'll maybe return to that in one of our subsequent lectures, but I want you to focus more on sodium reabsorption, the differential roles that luminal membrane proteins play in sodium reabsorption, and the important differences in water permeability that enable us to maintain water balance. So next lecture, we have a case of the day. We're going to start to talk about how the kidney regulates and fine-tunes sodium reabsorption. And then subsequently, we'll look at how the kidney fine-tunes and regulates water reabsorption. So we'll talk next time. Thank you.